Hi, everyone. Welcome to the 200 seminar series. I am delighted to introduce today's speaker, Professor Wen-Tso Liu. He's an Arthur Norman Endowed Professor in the Department of Civil and Environmental Engineering at the University of Illinois at our Urbana-Champaign. He does a variety of very interesting research projects under the umbrella of environmental microbiology. More specifically, more recently, his work has focused on the microbiome of water. So all things connected to microbiology of water that describes, different types of ecological roles of microbes in watersheds, drinking water treatment systems, wastewater treatment, water recycling and reclamation systems. And it's really important to, the science is important, the quantification is important, but just it takes a while for it to sink in. The roots of our discipline, environmental engineering, are in public health. And so the whole point of our discipline was to get microbes out of water. And disinfection is still a very important component of water treatment, but we are now trying to understand various other aspects of beneficial roles of microbes in bioremediation and biological waste treatment processes, but also the whole microbial ecology of natural as well as built systems that deal with water. And this thing's becoming much more important with advances in microbiology and molecular ecology and appreciation of that with climate change and everything. So it is very timely that Professor Liu agreed to talk with us about some of his latest research. Really interesting. I actually have never heard of microbial immigration. So I can't wait for us to hear and understand and learn in his own words. So um, please do join me in welcoming Dr. Liu as our seminar speaker today. Thanks, Wen-Tso. The stage is yours.
Okay, thank you for the introduction. So I have to say, actually, since when I was a child, actually, I learned UCLA, the name, probably one of the first school I know in the US. It's very prestige. And I was wondering, actually, maybe one day, actually, I would physically visit the university. But at least actually I get an invitation this time to actually interact with you guys through online, and I hope to visit you again in the future.
Definitely, yeah.
Okay. The title is Quantifying the Effect of Microbial Immigration in Engineering Water Systems. So immigration is the key word, and maybe for you, actually, you can just have premeditation about, say, immigrations for human being. As just through a country as a tourist for a few days, or maybe as an immigrant actually for a few years, or maybe through decades of time or century of time, and ask actually what's the impact after this immigration occur. And of course, the occurrence actually can happen in very different way, right? It can have because of natural disasters, can because of war, things like that. I'm not going too deeper on that as well. So this work actually, I have to say, is sponsored by multiple agents from the DOE, joint, it's also collaboration with Joint Engineering Institute. And also a lot is from British Petroleum and also the PepsiCo actually. I didn't put it on here. Also, it's been a collaboration with many collaborators throughout the world, and also with many utilities actually from 4 different continents. But last I have to say is that the main contributor actually is my students, particularly Lan Mei, who has just finished his PhD last year. And this is pretty much all his PhD work, and plus another efforts from Jin-Ha Kim as well. So I start with this urban water cycle, and actually urban water cycle is a perfect engineering systems, water systems, and it's used to sustain the growth of the human society actually. So what it does actually, it actually use a fairly good drinking water treatment plant to produce water from the source water. and then delivered to individual household. And after that, once the water is used, actually they are collected to centralized locations and treated at this wastewater treatment plant. To a standard is meet the discharge permit, and the water goes back to the source. All right, and this keep going on and on and on actually. But from the scientific perspective, actually, we can look at this actually as so-called continuum of water metrics, different water metrics that are connecting together. And then actually the water metrics just consist of first the water, right, pure water. And second, actually, you're going to see different gradients of chemical throughout the entire cycles at low and high concentrations as well. And also some abiotic particles, sand particles, some things, small nanoparticles we don't see with our. The last one actually is we seldom mention it a lot, actually is micro. Micro also move throughout the entire cycles as well, for many times actually. So let's take a closer look at the drinking water treatment plant. And we know actually the source water actually contains microbes. And once they actually go through the drinking water treatment process, actually, most of them actually are removed, but some still remain actually then ended up in the distribution systems and get out at the tap water when you open up. They also microbe actually seeded from the biofilters if you have one in the process, and they're going to seed the microbe into the distribution systems as well. They will come out from the tap as well. Another source actually is when we get to the premise plumbing region, the waters can flow and stay, and ended up there are going to be microbe actually growing on the surface of the pipe. We call them biofilm. And actually, they also can participate in the bulk solutions and then come out at the tap actually when you open it up as well. So on average, actually, you're going to get about 1,000 to about 10 to 5 cells per mils of microbes. Actually, depends on where you are in the States. It's the same with wastewater treatment plant, and in this case, actually, I'm going to use the one actually at the Urbana-Champaign, which is they use treatment filter and activated sludge as the secondary treatment, and followed by this nitrification tower to remove nitrogens. Okay. So in this system, you're going to see actually, again, the micro move in different way actually at different scale actually. Some can be a cyclic movement as well. And some can actually expose to different environmental conditions as well. So all of this actually, we call this as microbial immigrations by definitions. And then this actually defined by this scientist is by Bell, and it's a process which microbial individuals being added to a local community from the species pool of the meta communities, and which consists of set of local community next-by, and they actually physical link and by immigrations and can exchange colonies of multiple species. Okay. So this phenomenon actually is ubiquitous, both at the macro as well as micro scale. And this happens both in natural systems as well as engineered systems. And the difference for the engineering system from the natural system is the movement is a lot more controlled and also directly manners compared with nature system. So let's actually close look at the microbial immigration in the engineering systems where you can see, you're going to see actually the flux actually is going to be varies, depends on which system you're talking about. It can be very small in the drinking water treatment system. It can be significant higher actually at the wastewater treatment. The movement can be continuous, also can be semi-continuous as well. And the organism is going to experience different kind of redox potentials. And the outcomes when they move into a downstream environment actually can be different. They can be positive input to the downstream community, it can be also negative input or maybe just neutral as well. And the impact actually can be both directly and indirectly. Say it can be indirectly surplus carbon source as well. And maybe also can be a negative indirect impact because of public health as well. So it is actually very important to evaluate how microbial immigration will have impact on engineering water systems. So first, we actually would like to know to what extent they contribute to assembly of downstream community structures. What are the types and abundance? I mean, who are they and how much are they? And also to what extent actually they contribute to the ecological function in downstream process. And the last actually is, do they really help the performance of downstream process? So this is really big questions we like to answer. Throughout the years, actually, there are quite a few methods developed to look at microbial immigrations in engineering systems. And the first one is simply just by looking at the shear species actually between the upstream and the downstream community. And usually we represent this as a so-called Venn diagrams here. Second actually is so-called microbial source tracking. This actually is a function provided by this high throughput DNA sequencing pipeline, so-called chiming. And it actually uses a statistical approach to estimate what's unlikely the source of the populations in a sink environment or a sink communities actually. And the third one is actually is use this so-called neutral community model or neutral theory actually, which they simply calculate the so-called this immigration probability M actually. When the number is approaching to zero, suggests the immigration is very strong in a given environment. And when it approaches to zero, suggests the immigration is actually quite low. But if you look at three methods here, they're all very qualitative. Actually, they don't provide quantitative analysis. So it's a question whether can we actually quantify these microbial immigrations. And for that, actually, my student actually has developed this so-called ecogenomic-based mass balance approach. So to elaborate this approach, I'm going to use this chemostats as a model. And so for that, actually, we actually can estimate for a given organism X and how much they actually enter the chemostat and how much of them actually remain in the chemostat and coming out in the chemostat. And by simply applying a mass balance, and we can write our equations on this for this organism X actually. If we're assuming this is chemostat has to reach steady states, you actually can simplify these equations and be able to rearrange these equations, have the specific growth rate here, mu max, mu X stands for the organism X, is actually equal to this 10, which is 1 over SRT, minus the percentage of organism X coming in and also divided by the reactors. And also divided by HRT and also by the biomass, total biomass coming into the systems, divided by total biomass in the reactors. And this becomes a really simple term we can actually measure. What we can do is SRT and HRT and total biomass you can measure from the operational data, right? And for this percentage coming in and in the reactors, we actually can leverage on this high throughput sequencing technology. And with knowledge, we can actually have very good estimations of given population out of total population right now. So we can calculate the specific growth rate for organism X. We can actually apply to every single organism. And then we can apply it to these diagrams. So I'm going to walk you through these diagrams here. So the x-axis actually is the growth rate here. Y-axis is represent abundance in upstreams. What's the population coming in? And the bubble size represent the abundance of the organism inside the reactors here. And you're going to actually reach a maximum growth rate is equal to 1 over SRT. That's actually how high actually the mu max can go actually. So with that, actually, we can see here, if the organisms present on this side, actually, we can say they are actually dominant population in the upstream, but they are kind of dying off or they are lysed actually in the downstream environment. They are not active much. But if they're actually present on this side, actually, we can see they're likely active populations. Maybe they're actually seeded from the upstream environments. However, if they're actually present here, actually, we likely suggest they are, this environment actually probably very unique to them. It's a niche for these populations actually. So if you compare these methods with the other three, actually, the EGMP model actually can quantitatively estimate the individual abundance and activities. It actually can also deal with multiple upstream environment and multiple downstream environment. You actually can quantify microbial biomass as well. But actually, you need assume actually this is a steady state conditions or quasi steady states. And you actually have to repeat this, taking a sample throughout the study to achieve this as well. So I'm going to give you 3 examples here actually. And first one is on anaerobic digesters. And I'll come back with the other 2. Anaerobic digester is actually perfect systems for microbial immigrations. Because the feed population coming as a feed biomass is coming in, actually, they are degraded to biomass by gas like CO2 and methane gas. With the collected efforts from different group of organisms like hydrolyzers, fermenters, syntroph, and acetogens and methanogens. Right? And we know actually the biomass coming in, the diversity is quite high. All right. And loading actually is the flux is also quite high. Often the digestion efficiency is not 100%. So you will ask what happened with the remaining populations? Do they actually play certain important role in anaerobic digester or not? Or do they become something else actually? So it's I'd like to answer that. So to answer all these questions, the student actually, so we have collaborated with the STICME treatment plant at Chicago. It's one of the largest wastewater treatment plants. So in this process, they receive multiple sources of the feed from primary and secondary sludge, from a wasp from another treatment plant, and also the sludge from in-host digestion tank as well. So they all enter this holding tanks mixed, and then fed into 24 anaerobic digesters. And then after that, actually they are dewatered and ship out for either composting or landfill. So actually we took 3 of the 24 anaerobic digester along with the feed and sample for a period of 4 weeks. We then actually take the biomass, extract the DNA, sequence the 16S, and determine the core populations. And then later on use the core population to determine the growth rate applying to the EGMP model. And we also, at the same time, we actually asked the operational data from the treatment plan as well. So this is after the sequencing data, actually, we were able to assign them to different so-called operational taxonomy units or different microbial populations. We plot them against the occupancy here. And so you can see the distributions of any given populations with their occupancy or occurrence throughout the sample. We also plot their relative abundance here. So based on this, we found out there are about 375 populations they present in all sample taken. And they represent about 80% of the total populations here. If we make another cutoff and say, let's just look at populations greater than 1%, they end up to be about 74 OTUs here. And we call them, we kind of define them as our core populations here. And we apply it to this EGMP model, and this is the result. So with the, again, the growth rate at the bottoms, the abundance for the upstreams as y-axis, and the bubble size as the population's abundance in the reactors. The student actually look into individual O2 in terms of who they are, what are the population they are associated with, and identify them as whether they are so-called aerobic or facultative anaerobic, denoted as yellow, or whether they're aggregate anaerobic, which is red here. And you can see really nice distribution here. Suggests all this is a population from upstream environment. And this actually is a really key population making the digestion work here. I can point out a few here. Zooglea is a really known flood-forming microbes, actually. It actually contains quite a lot of EPS. So maybe it's this another reason, actually, it tends to be difficult to digest the zooglea. It's just a speculations here. Again, I can speculate Thyra, which is a known denitrifier. Maybe it can survive a little bit better under the digester because there's still some slightly presence of nitrates in the digester in the beginning as well. We actually then find out this population are key population, and likely they actually enriched from the seedings, actually, from either very beginning or constant seedings, say, for example, from Inhofe tank here. We also find some really unique population to digesters, and they include some organisms we know they are completely anaerobic here as well. With that, actually, we can actually estimate in upstream environments like activated sludge, how much populations actually is digested based on their growth rate, and how much is actually still remain, how much actually become active because they have a positive growth rate here. And in that case, actually, we can estimate it's about 82% of the population in the activated sludge actually has been digested in the digester as well. So overall, you can see here the immigration is actually very strong in the digester, and primarily due to, I think, the input as a carbon source. So I'm going to bring you to another second case. In this case, it's combinations of USB stands for upper floor anaerobic sludge blanket reactors plus the activated sludge process. So it's a reverse process of activated sludge plus digester. Now it's actually have anaerobic upstream and aerobic downstream here. And this system is to treat this purified terephthalic acid wastewater. Maybe most of you don't know what is terephthalic acid wastewater. We call it PTH. So PTH is one of the highly synthesized chemicals in the world right now. It's probably on the top 10 list. It's actually derived from the crude oil. Actually, part of it is produced as a xylene, and xylene actually used to produce PTH here. And PTH is used as raw material to produce all the materials we're using daily, actually. And the markets actually represent, actually, it's up to almost like 1,000 billions now per year. So it's huge markets here. So PTH is produced through this weight oxidations with xylene as the raw materials and end up with terephthalic acid. And throughout this process, it's going to produce a lot of wastewater, which are mostly aromatic compounds and just a little bit like acetic acid and others. And this has to be treated because the concentration tends to be really high, about from a few grams to 10 grams. And the volume is quite huge. I think it's equivalent to about 20 million populations per year. That's how much volume is produced. So it's treated with these combinations of USB and aerobic system. So you actually have an aerobic system coming in with a concentration of about 3 to 6 grams. 90% of it is removed, you end up with about 0.3 to 0.6, and they're actually further treated by activated sludge process. To a level actually is less than about 30. And right now, actually, the company they actually take it and actually treat it again with the membrane and produce a reusable water. So right now. So we asked actually is how much impacts of immigration we have from the USB to the downstream aerobic systems here. So we actually use a full-scale systems with British Petroleum. And they actually have this configuration with the USB followed by 3 parallel activated sludge process. And we actually have 6 different sampling times. And at all these different locations, we sample it. And again, we follow exactly the protocol. We extract the DNA, we sequence the 16N sequence through the high throughput sequencer, analyze the core populations, and apply into the EGMP model, together with the operational data as well. So you can actually establish the model for this, which is slightly different from the chemostats. But it's likely the result is pretty similar searching. So I'm just gonna walk through with, so you're gonna have this mu-max again with a little bit more complicated equations. Again, all these parameters can be measured either through operational data or through sequencing data as well. So then we actually can apply to this figures, but in this case, I'm gonna change a little bit. Only have the growth rate at the bottoms and abundance of activated sludge in the y-axis here. Because the input actually is really low to showing up, so we are not able to show it really clear. So we can see actually, you can see actually population located on this end is tend to be more active in abundance here. Okay, so this is what we see here. So we're going to have a lot of population here with a few are more abundance and the others actually are relative low in abundance, but most of them actually are active. There are very few inactive populations here. If you look at these diagrams, a pie diagram here, the active population represent at least about 98, 90, I think 97 to 98% of the total population. In contrast to the digestion we just look at here. So in summary, you can say the immigrations from the upstream USB reactor to the downstream ashes, activated sludge ashes is relatively low here. So what my student decided to do is to further improve this with so-called machine learning algorithms. And it's really interesting the way he presented. I'm going to skip these 2 slides, actually, I don't need to use. So it's using machine learning. So what is machine learning? He gave this example saying he actually can establish a relationship between the salary and yield of educations by getting enough data sets and then train them actually and establish these so-called associations or so-called regressor between the salary and yield of educations. And then you can go out to sample, randomly sample a few samples and ask actually the people their salary and the yield of educations. And they will ask whether the predicted actually, observed actually really corresponding well with predicted. And if actually the prediction is pretty well actually, then actually with observed actually, then actually you can use this quite well to predict something later on for the other people as well, right? Or you actually can, you can look at another parameters. Let's look at salary and length of the name actually. And my student's name is Lanmei, has 6 letters, right? So if you look at that, I mean, we actually see actually, usually you get really poor correlation between the observed length and predicted length based on names actually. And because if it is true, actually, the prediction is pretty well. I mean, you know, some Indian students, their names tend to be really long. They're probably more than 20, 30 letters, right? And my student's name is only 6 letters. So fortunately, say this was not the case for him. So you then apply this to the study actually by correlating community structures with the temperatures. And train them, establish regressors and test a subset from the study and see how good the relations will be. And this is what he observed by taking entire communities and and predict together with the temperatures, you see a pretty good correlations. But if he eliminate those immigrants, but just look at all the active populations, you see the correlation is getting a lot better. And this actually further support his observations that the immigration effect is quite low in this study here. You actually can apply to all the different population set by looking at the entire community and also the immigrant-free community. And you actually have a lot higher accuracy for predictions based on active community. So this actually become a really good tools for wastewater treatment plant in the future. So once they have is definitely enough so-called big data, they actually start can predict their own performance based on the operational parameters as well. Okay, so I have one is very strong immigration and the other one is very low immigration. And this way I look at another scenario, which is WAS, waste activated sludge degrading fermenters. So often actually what we do is we have the WAS and digest with digester and produce by gas. But with the kind of newer thinking right now, is just to use WAS and actually fed into fermenters and produce some precursor for high value added products. And actually they can be produced as a PHA and actually biofuel as well. So to make fermenter work, and there are a few operational parameters going to influence the performance, and including the inoculant heat shock, pH value, SRT, and others. So our study is primarily to look at the SRT here. So what we do is we take the WAS, we took the WAS from the PTA wastewater treatment plant, and then fed into a chemostat, which has inoculant from a local anaerobic digesters, which is heat shock first and operate about 5 degrees of pH 5 and 39 degrees C. We then actually use this moderate reactors and subset to quite a few semi-batched reactors and look at the SRT effects from 37, 38 days down to 2.3 days by changing the feeding volume and frequency here. And this goes on for about 50 days or 60 days. During this process, we, of course, we also monitor by gas production and DFA, and also by biomass concentrations. And the sample taken during each fed, the biomass sample was actually analyzed again for their extracted DNA, analyzed for 16 sequencing, and then apply into the EGMP models. So you can see one of the results from the SRT is about 38 days. And this is the diagram I showed you in the very beginning. And you actually can generate the same diagrams for every single SRT here. There are too many data here, so we decided to kind of simplify the analysis. So let's look at the top, the most top abundance populations. And we look at 4 different groups here. So we look at actually most abundant OTU from, they are known as so-called syntroph or known as methanogens, and also OTU which has a positive growth rate, and also OTU has negative growth, and find out who they are actually later. So these 2 diagrams you see actually, again, on top is the syntroph and bottom is the methanogens, and the x-axis is the growth rate, and y-axis is the net growth rate as well. It's SRT versus, so it's SRT versus, more of SRT versus net growth rate. So for methanogen and syntroph, what we see actually, with the decrease in SRT, we see the abundance in general decrease for both, as well as the diversity. They tend to be either washed out or become as a negative growth rate. Only a few actually still remain. Particularly, there are some still really stubborn methanogens still remain at really low SRT. But syntroph is almost gone here actually. right? So it's really clear because the effect of SRT has very strong effect on the central because they decouple the relationship with the messengers here. We further look at those with positive growth rate, and you can see here they have 100% linear relationship with the one over SRT, suggests they are really active populations here. And the other ones actually is the negative growth rate suggests they are likely washed out throughout the population, or they are fermented. That's what I meant actually here. And what we actually so we decided to look into what are the population they have associated with, what are the phylogenetic name of those population they have associated with. And on top, we found out all these populations are associated with member of fermenters here. And this actually further evidence actually, yes, fermenters are doing the work in the reactors here. And at the bottoms, these are mostly the dominant population from the WAS populations here. So it's really clear you can identify who is doing what using this approach. We then actually correlate with the operational data. So we can see here with the decrease of SRT, we see actually a decrease in the methane productions here, and also a slightly decrease, of course, in solid reductions here. But we see a huge increase in volatile fatty acid productions. So the question is, if the industry actually running this wastewater treatment plant, they decide to use water to produce fatty acids and also trying to achieve good solid reductions, what SRT should they use? And the reasonable range is probably fall into this range here. They still can achieve pretty good SRT, I mean, the solid reductions and also VFA productions here. So this is pretty much the outcome from that study here. So you actually just saw 3 different cases here. And I have to say, actually, there's some different caveats with the EGMP, of course, has great potentials. Then let me walk through with you actually. For this EGMP model, we actually measure this abundance of a given organism with based on the DNA extraction, PCR, and sequencing. All these actually have bias associated with them. Also, copy numbers of RNA gene sequence as well, right? And to estimate the total population, we use volatile suspended solid here. It works well with pentonic cells or with flux, but it may not work very well with granular or biofilm. It's really hard to estimate the biomass, total biomass in systems. So maybe there are other surrogates actually we should explore in the future. And also biomarkers here, we use the 16S. Maybe another way we can use this by looking into specific functional genes. If we're interested in a very specific group of organisms, or maybe use housekeeping genes here as well. And if actually you have sufficient money, actually, and maybe in the future, next few years, actually, this won't be an issue. We can actually direct sequence the entire genomes and do the apply into the EGMP models for that. This allows you to look at even at species or strain level resolutions. How about viruses? This is something actually I've never mentioned so far. And with the COVID happening right now, this maybe is also another great potential to apply into it as well. Together with the other bacteriophage is so important in the systems. And my student had demonstrated actually you can actually combine with AI and actually require a big data set. And this really can help the process operations in the futures as well. So we see there are great potentials. And I think actually it has been applied in quite a few cases from our group and some other researchers groups here as well, including wastewater treatment process for activated sludge process, anaerobic digester fermenters, MBR, membrane bioreactors, also granular reactors, and other type of reactors if you're working on it, that would be great. You also actually work with drinking water treatment systems here as well. And let me give you two examples here why it works well. So the first one is premise plumbing, actually. As you know, actually, when water actually move into the premise from plumbing, actually, the water most of the time is stagnant and then move, and then stagnant for a long time and move, actually. And through this cyclic events, actually, we tend to actually have biofilm develop on the pipe wall because there are organisms coming in together with fresh water here. So actually, you actually can model actually microbial growth in the box solutions and also in the biofilm growth and also the cell detachment from the pipe wall. You actually can couple with chlorine decay, looking into in the box solutions and how they affect the pipe biofilm growth as well. And by combining this model and applying with the EGMP, I think we should be able to carefully look at the microbial immigrations in these systems better than we have right now. Another system is a rooftop water tank. Have you seen one like this? If you take a train, maybe in Japan or maybe in China, just you're going to look out the windows. You're going to see many tanks on top, sitting on top of the roof. You come to Chicago, actually, you're going to see this the wooden tank sitting on a lot of buildings here. This is a perfect chemostat with a semi-continuous or continuous mode. It has the growth in the box solutions. It also has growth in the biofilm as well. So if you model it correctly, you also can see how the immigration takes place in this system as well. So the ultimate goal for us to look into microbial immigrations to answer a few key questions. Definitely we'd like to know actually what are the most important microbes and what do they do in these engineering systems. We also want to see actually whether can we understand this and be able to improve the engineering water systems performance, and maybe also be able to protect the public health in the long term. Actually. And so for that, I would say actually, if you're actually working on some systems, or you have studied so far, you're planning to study, think again whether this can be a good case for you to apply this EGMP model into your systems. If yes, that would be great, and I will look forward to your future work in an article coming out as well. So my last words, thank you, and if you have any questions, and also you have 3 more weeks, so you can enjoy your coming summer break as well. Thank you, everyone.
Thank you so much. This was very interesting. I thoroughly enjoyed it. We have some time. So we can open it for questions from the students first.
Hi, Professor Liu. This is Yifan. I have a question about your presentation. It's a really good seminar. And I have a question about the last slides of case 1. So
the case 3 or
case 1.
Case 1.
Oh, yeah. Case 1.
Yeah.
Last slide, case 1, then you go back to.
There's no like the number, the page number for the slide, so I'm not sure which will be the number.
I think I have it. I just need to let me see if I can make it work.
Yeah, there's students.
This one.
Raise your hands or type in your questions in chat and I'll ask them for you. You can ask, yeah.
So you mentioned the immigration is due to probably the carbon source. So I'm just wondering like what carbon source will be in this reactor and what kind of carbon source will lead to the immigration?
So I said the impact can be direct and indirect. So look like most of the population participate so-called in an indirect way because their biomass becomes the food for the population in the digester to grow on. But there are some actually that you can see that some population, they actually do have a positive growth rate. And they actually probably are serving as a seed and become a dominant population in this digester. And likely we can cause bio-augmentations as well in that sense.
Thank you. Thank you.
Yeah. Good questions.
Yeah.
Hey, Professor Liu. This is Rin. I'm a postdoc in Shelley's lab. I did some similar work, microbial ecology. I'm wondering, so in your model, you most focused on the 16S sequence, so on the taxonomic level. So have you ever thought to quantify the function of the community?
Yeah.
And whether this immigration has some relationship with the function?
It's excellent questions. I've been actually asked quite a few times on this as well. And I've been very cautious about this because we actually took granted too much about 16S sequence. We think 16S sequence is almighty. It can do everything. It can tell you who they are. It can tell you what they are doing. And the trick is most of the time 16S does not tell you exactly what's the functions of organisms. On this, actually, you are pretty much sure they are associated with very defined lineage with a lineage actually with very defined function like nitrophile or like a central carcinogens. I probably would start to look into their functions. If they are actually associated with lineage, say Bacteroides, Bacteroidetes or Firmicutes, I actually cannot tell exactly what they are doing. So I want to be very cautious about how we use 16S to predict functions in our study as well. But what you can do is, that's why we want to move on to, why don't we try specific gene markers, right? Or maybe we can eventually apply the whole genome, like so-called MAG, metagenomic assembled genomes to look at their functions later. But I believe this still take a few years to evolve because of the technology. And also, I think it's pretty much the sequencing limitation here in terms of the cost, I believe.
Then I have a follow-up question. So do you think it's worth or it's necessary to run some experiment by using the pure culture? For example, here you have some cool microbiomes. So it's worth to run the pure culture to test the microbial traits.
Excellent. This is what our thought was about 2 or 3 years ago. I actually had one master student working on it. It's kind of was heavily interrupted by the COVID. So we actually are still working on, we cannot, otherwise we probably will be able to share some good data on that actually. Yeah, we're asking ourselves, what happens if we add just pure cultures? Can we see this, can we apply this model into the system and prove this is working as well? But feel free, if you're interested, you can jump in and work on it as well. I think there are different organs and it should be tested on this model actually.
Yeah, thank you.
Yeah, good questions, both of you. Anyone else? Wen-Tso, if I may follow on with Rain's question about, the taxonomy versus function, I do think if we, and not to take away how important it is to understand the phylogenetics, but we might be missing out on horizontal gene transfer that happens in these complex communities, which can, enhance or take away function, but would not be reflected in the 16S or 18S based assessments.
Right. Horizontal gene transfer is too difficult. You actually don't know who is the host, who is actually the ancestral cell and which is the recipient cell. So this actually poses a really big question. But maybe we actually can carefully design an experiment to look at the transitions.
Yeah.
If actually, so there was some discussion with my students in the past, actually, if we're able to introduce an organism which contains a specific plasmid, we know actually we can put a biomarker on.
Right.
And we know actually by introducing into a system, we can start to see how it transmits through the horizontal gene transfer to other population throughout time, and see who become the recipient. This actually, but it's still so challenging for this, because we end up with a recipient. You still don't know who is the recipient, because when you try to sequence them, you are not able to sequence them together with 16S. Even if you want to sequence the whole genomes, it also does not tell you who they are as well, because they are separate. I mean, the plasmid genome and the chromosome actually are separate. So that was our discussion ended there actually a few years ago. But again, Shelley, I welcome you to pursue this session. This session will be, I think, very, very interesting if you are able to quantify the horizontal gene transfer in the system. Yeah.
Yeah, that's something we tried to do many years ago for an in situ bioaugmentation project, because that would have implications on how often do you need to augment. So immigration in that case for soil and groundwater remediation. And if there is even benefit to putting live microbes rather than labile DNA.
Yeah.
And everything else. Well, Rain had just started as a PhD student at that time. But, yeah, there are other things, but that's a question that I've always asked, identity versus function. I mean, do we really care about names of everybody who works at Apple, or just what Apple devices can do for us, or what their roles are? Not to say that that's not important. Just a balance between identity and function of the microbiomes.
And the third case, as I mentioned about, it was very clear by using the conventional way, just looking at abundance, we actually were not able to tell who is active there, who are active there. But using this EGMP, we clearly can tell who are the active fermenters right away.
Exactly. Yeah.
Virus is another big hurdles, because virus actually copy numbers increase enormously after the lactic cycle as well. So they actually also create another issue to model that for us.
Yeah, no, lots of work remains to be done in all things omics. So yeah, it was good. We learned a lot. Thank you so much for your seminar and your time. Any other comments? Otherwise, we'll just thank Professor Liu.